Good morning or good afternoon, everyone. I'm Julia Caro, editor here at Genome Web, and I'll be your moderator today. Today's webinar is entitled Comprehensive Tumor Characterization for Immuno-Oncology, Clinical Trials and Cancer Research. The sponsor of this webinar is Personalis. Our panelists today are Dr. Richard Chen, Chief Scientific Officer at Personalis, and Dr. Michael James Clark, Bioinformatics Applications Manager at Personalis. You may type in a question at any time during the webinar. You can do this through the control panel, which usually appears on the right side of your screen. Click on the Q&A box in the upper right side of the control panel. When you click on Send To, please select All Panelists. We will ask the panelists questions after the presentations have concluded. Dr. Chen, please go ahead with your presentation.
Terrific. Thank you, Julia. Well, welcome everyone, and thank you for joining us today on the webinar. Today, Dr. Clark and I will be discussing Personalis' NGS sequencing and interpretation services for cancer, with a specific focus on our ACE Immuno-ID solution, which we are launching to support immuno-oncology clinical research and clinical trials. Next slide. We're going to start with an introduction to Personalis. Then I will briefly introduce immuno-oncology and some of the key NGS needs in that area. Then I'll turn it over to Michael, who will be discussing our ACE Immuno-ID solution in some detail. Next slide. We won't be able to cover everything today, so I encourage you to come see us at ASHG. We have 3 terrific platform sessions there covering a variety of topics. Tumor normal versus tumor analysis, which is a hot topic of discussion these days. RNA and DNA integration, which is very relevant for immuno-oncology. And our hybrid exome whole genome assay approach. Next slide. We also have 2 workshops served over breakfast and lunch, one on cancer and one on inherited disease testing. We also have a number of posters and a booth, so please come by to find out more. Next slide. So a little background about Personalis. We are located in Menlo Park, California, near Stanford, where many of our founders are from. We were founded with the mission to build innovative, comprehensive, high-accuracy NGS and sequencing and interpretation services focused really on 2 major areas, cancer and inherited disease. We have a broad set of research and clinical services in these areas today, with customers in over 10 different countries spanning pharma and biotech, hospitals and clinicians, government, academic centers, and researchers. Our CLIA lab and high-performance computing center has delivered thousands of NGS sequences and interpretations to date. Next slide. To pursue our mission, we brought together a world-class team with deep academic and commercial experience spanning NGS informatics and diagnostics. Next slide. Starting with the founders, Mike Snyder, John West, and the others, they recognized early on that going from DNA and RNA to a clinical interpretation in NGS is a very complex process fraught with significant accuracy challenges that really require novel solutions. This includes things like sample prep, where there are challenges with FFPE, small sample amounts, RNA and DNA quality, diverse sample types, sequencing, where there can be large gaps in medically important genes. This is something we'll talk about more later. Informatics challenges with calling indels, CNVs, fusions, especially at low allele frequencies, and also reference issues that can lead to blind spots in analysis. And then there are content and clinical interpretation challenges with variant classification, for which the guidelines are still being developed in cancer, and then accuracy of the database annotations themselves. So Personalis has tackled many of these issues head-on and developed novel technologies to solve them. Next slide. So that technology has come together in our accuracy and content enhanced, what we call ACE, clinical NGS platform, where we haven't just pieced together off-the-shelf solutions, but really innovated at every step of this process from sample to interpretation. So what's key here is that we take an integrated approach to developing our solutions. So solving these hard problems often requires development and optimization at multiple steps in the process simultaneously. So for example, accurate detection of CNVs and fusions involve developing novel methods for sample prep, sequencing, and informatics that all work together. And so thereby, by taking an integrated development approach, we're able to maximize the accuracy across the entire process for specific applications in cancer and inherited disease. Next slide. So the ACE platform itself forms the technological and accuracy foundation for all our products spanning clinical research, clinical trials, and clinical diagnostics. And we have products both in cancer and inherited disease across all these different areas. Now, we're not going to have time to go through all our products today. Today's focus is really on the next slide, ACE Immuno ID product, where we are launching this at ASHG. This is a product focused on immuno-oncology, clinical research, and clinical trials. Next slide. So Dr. Clark is going to walk through the biology of immuno-oncology in much more detail, but I'm just going to give a brief introduction to immuno-oncology for those of you who are new to the field. So immuno-oncology is a hot topic for good reason. In a nutshell, immuno-oncology is focused on harnessing our own immune system to treat cancer. And this concept has been around for a long time, actually starting with Ehrlich in 1909, who first established that the immune system can search and attack transformed cells. But it's really only in the last few decades that the molecular mechanisms themselves have begun to be elucidated. And a key part of the molecular story are the immune checkpoints, such as CTLA-4, PD-1, LAG-3, and others. And they're just a normal part of our body's regulatory mechanisms for keeping our immune system in check to prevent damage to healthy tissue. However, it turns out that some tumors have gotten good at upregulating these ligands for these checkpoint proteins, thus enabling them to evade the immune system. And so new inhibitors of these checkpoints, such as ipilimumab, nivolumab, pembrolizumab, and others, really raise the potential to mount a sustained immune response to the tumor. Next slide. And that's what's incredibly exciting about this area, that these new checkpoint inhibitors dramatically improve outcomes in some cases. But not every patient responds equally, and not every tumor type responds equally. And so the question increasingly that people are asking is, why? What modulates the response? Is it the tumor immunologic microenvironment? Is it the expression of these checkpoints? Is it the broad mutational burden or specific neoantigen signatures? There are some terrific papers. This is a figure from one by Rizvi et al. At Science, published earlier this year, that show that there's reason to believe that there are predictors of response. In this case, they showed that the neoantigens and the burden of neoantigens can be predictive of response to immunotherapy. Next slide. So if you're looking for expression about biomarkers, or even just trying to characterize the broad mutational burden, what are some of the key upfront NGS needs? Well, one thing that is clear is that you need a broad assay. A narrow panel isn't going to get you there even with 50 to 500 genes. It's not sufficient because you're not able to see the broad spectrum of mutations in the tumor that you'll want to see for neoantigen identification. The other element is that the coverage needs to be complete and accurate. You don't want to be missing potentially important immunogenic epitopes. RNA-seq, incredibly important. DNA only is not enough. So when you're trying to filter down the scope of potentially relevant neoantigens, you need to be able to know which ones are being expressed. You want to be able to be confident in your somatic calls. This is especially important if you're relying on the neoantigens for identifying potential epitopes to target in vaccine development. So things like tumor normal analysis can really be important to improve specificity. And then finally, if you're going to use this information for clinical trials, the assays and informatics need to be robustly validated. Next slide. So if you look at many of the options, the clinical cancer sequencing options out there, a lot of them come up short for the needs of immuno-oncology. Typically, these solutions are focused just on the tumor, not tumor normal analysis. They just provide DNA sequencing versus DNA and RNA. And the panels themselves are typically quite narrow in scope. Now, there are exomes out there available, but those exomes oftentimes have accuracy issues, sequencing gaps that can occur that can be problematic if you're trying to identify neoantigens. Next slide. So with our ACE Immuno-ID solution, we've developed what we think is a comprehensive solution for immuno-oncology clinical researchers. At the core of our Immuno-ID solution is our ACE cancer exome. And we'll talk much more about that and why that's differentiated. We've also developed the largest cancer panel on the market, the ACE cancer panel, with over 1,300 genes, 200 microRNAs that are suitable for certain immuno-oncology applications. For both these assays, we're offering not just the DNA sequencing, but the RNA sequencing paired with the DNA, which is, again, critical for immuno-oncology. And we're doing not just the tumor-only analysis, but we're doing the tumor normal analysis to increase the specificity for identification of somatic mutations. And by having this broad survey and characterization of the tumor, we can characterize SNPs, indels, and CMVs, as well as broadly characterize fusions and gene expression through the RNA sequencing. In addition to our sequencing and kind of sample prep capabilities, we have strong annotation databases, over 40 databases. For clinical trials, we recognize that there's need for flexible reporting and data delivery options, and we have that as well for customers. So this speaks to the comprehensiveness of our ACE Immuno-ID solution, but the other part of the story is the accuracy. And so the next slide, please. So in terms of accuracy, we bring the accuracy of the ACE platform to the ACE Immuno-ID product. So starting with sample prep and sequencing, where we fix biases in the sequencing and sequencing gaps, to the robust CLIA validation of our assay and informatics pipelines, we are adding a lot with our ACE platform to increase the overall accuracy and comprehensiveness of the solution. So this is the full scope of our ACE Immuno-ID solution. I'm going to talk more about, Michael and I are going to talk about the ACE Immuno-ID solution in more detail at this point. Okay, next slide. Okay, so as I mentioned, I'm going to start by talking about the ACE assays. This is really at the core of the solution. There are really two parts to the ACE Immuno-ID solution. Really, the core is the ACE cancer exome and the ACE cancer transcriptome. But we also have the ACE cancer panel as the largest cancer panel available that also is suitable for certain applications. The ACE cancer exome and transcriptome have been specifically augmented to improve the accuracy and performance for over 1,300 cancer-related genes, 200 microRNA genes, and over 6,000 other medically important genes. And of course, as an exome that covers all 20,000 genes in total. Another option is the ACE cancer panel for DNA and RNA. Again, I mentioned this is the largest cancer panel on the market with over 1,300 cancer immuno-oncology-related genes, as well as the 200 microRNA genes that I mentioned before. And it covers some of the important immuno-oncology-related genes and is viable for some specific immuno-oncology applications. So all these assays, the key here is all these assays are augmented with our ACE technology. So I'm going to talk a little bit about that now. Next slide. So what are some of the problems with standard sequencing approaches today, and how do we solve them with ACE? Well, the first misconception is that an exome is an exome and a panel is a panel. The reality is that conventional NGS assays can leave large sequencing gaps in key medically important genes. So here's just one example of this. This is STK11. It's a very important cancer gene associated with the onset of intestinal polyposis and the risk of developing different types of cancers. It's been associated with lung adenocarcinoma, for instance. And the plot here, the blue plot here, shows the coverage across all bases, all exonic bases in this gene, all concatenated together, so you can see the coverage all in one plot. And so what you can see right away is that there are whole exonic regions and variants residing in these regions that would be completely missed. In fact, there are panels that actually claim complete coverage of STK11, but are in fact leaving out some of these hard-to-sequence regions out of their definition of the gene. So the point is that when you're comparing results from different panels and exomes, you really need to kind of dive into the details because people define these genes in different ways. And it turns out that these particular exons are difficult to sequence regions, high in GC content, and that's why a lot of folks will leave this out of the definition for either their panel or their exome. Standard exomes have this problem. This is true for panels. This can also be true for whole genome. The main point here is that these gaps can lead to misvariants, and in the case of Immuno-Oncology, missed neoantigens, and we'll show examples of that later. Next slide. So another misconception is that simply sequencing deeper solves the problem, and clearly this doesn't. So here are two examples, again, two medically important genes, RPGR and SDHD. And you can see these are sequenced to relatively high average depth, over 100x coverage, but you can see that there are regions, gap regions in both these genes that will remain poorly covered no matter how deeply you sequence. And this can be due to, again, various systematic biases, IGC content, segmental duplications, and other reasons. Next slide. So to solve these problems, we've developed our patented ACE sequencing technology, which we use in all our NGS exomes and panels that we develop to really achieve high sensitivity and clinical grade performance. So we recently published this in Genome Medicine, co-authored this with NIST and the Genome in a Bottle consortium, showing how this approach, the ACE approach, really enables much more uniform coverage and higher sensitivity, especially compared with other exome and even genome platforms. Next slide. So our ACE approach works through doing multiple optimized targeted capture paired with optimized sample prep specifically designed to fix gaps and the systematic biases found with NGS sequencing, standard NGS sequencing approaches. And the point here is that simply adding more probes doesn't solve the problem. You need to optimize, again, the targeted capture with the sample prep to really get at some of these issues. And so we do this, these separate targeted captures under different sample prep conditions, and then we combine the results into a single high accuracy data set at the end. And so I'll show you in a moment, sort of, at least visually, how that works. Next slide. So here are standard, so here in blue, you can see standard assays will kind of target some of the straightforward to sequence areas, and that's in blue. And with our ACE approach, using these multiple targeted captures, we're able to fill in some of the hard to sequence regions. So next slide. So you can see that this shows how it combines, we get the combined coverage from these different targeted captures that results in high finishing rates for genes. In other words, complete coverage over these genes. And we call this process gene finishing. Next slide. So back at the STK11 example, this is again the original plot with standard, showing the results with standard exome sequencing. And the next slide. The next slide shows that with our ACE approach, in green, you can see that we filled in many of the gaps that occur with standard approaches. And so you end up with much more uniform coverage. And the point here is that you won't miss variants in these regions. Next slide. And here's just some other examples. These are really important cancer genes, BRAF, NF1, TP53. And you can see, again, these will each have their own issues and sort of peculiarities and sort of which regions are difficult to sequence. But you can see that we've addressed a lot of those issues and really filled them in nicely with our ACE approach. Next slide. So, coming to the paper, I've shown some specific examples, but how does ACE improve quality overall? And so we don't have time to go through the entire paper in detail, but this is one of the figures from the paper that compares our ACE exome with the majority of modern exomes, as well as the 31x whole genome, PCR-free whole genome. So you can see that ACE performs the best in terms of achieving high coverage across coding bases. So if you take a look at the graph on the left and follow it up vertically from the 100% of bases covered mark, you can see that the number of exons the 100% covered to a minimum depth at 12G is significantly more than other platforms, including whole genome at 30X. So ACE covers over 99% of coding bases at greater than 20X, compared to 10% to 78% for whole genome and other exomes. Furthermore, ACE finishes over 90% of exons compared to 30 to 65% of exons for other exomes and 10% for whole genome. Next slide. In this figure, you can see that the performance differential is even larger in difficult to sequence regions. So in this particular case, we focused in on just those regions of high GC content. And you can see if you look at regions of GC content greater than 80%, we're able to maintain good coverage in those regions, whereas other platforms begin to fall off in terms of their performance. Next slide. The ACMG has recommended reporting on very specific, very important medically actionable variants. It's called ACMG 56. And so what we did was take a look at those variants and genes and look to see how well we cover them compared to the other exome platforms and also whole genome. And it may be a little hard to see here, but in green, the green triangles show our performance, which is really terrific across all these different genes. We actually stacked the deck against ourselves a little bit. We took the highest performing exome out of all the other exomes and compared that to us, and that's what's plotted on this graph. And you can see that even if you take the highest performing out of all the other exomes, that their coverage is all over the place and in many cases poor. So we compare quite favorably there as well as compared to whole genome. Next slide. Now, for cancer, if you look specifically at cancer-related genes and the number of cancer genes finished by other exomes compared to us, we're finishing 20 to 30% more of those genes. Next slide. So what's the impact of this quality enhancement on neoantigens or immuno-oncology? Well, here, this is just to hit home the point that these are some genes that have been reported to harbor immunogenic epitopes that are relevant, possibly relevant neoantigens. And you can see that these genes, again, have these gaps in coverage that need to be filled. And so if you're looking for these epitopes and you don't do this kind of augmentation, you're likely to miss some of these. The next slide also shows, again, these are genes that have been shown to harbor immunogenic epitopes, possible neoantigens. And again, you can see that the coverage can have gaps, and so that, you know, by using the ACE technology, we filled in a lot of those gaps and really giving you confidence that you're covering all the epitopes that are out there. Next slide. Okay, so I've talked quite a bit about the DNA sequencing assays, but the RNA-seq is actually as critically important for immuno-oncology. Now, Michael is going to talk in more detail about the application specifically to immuno-oncology, but I just wanted to touch on how our ACE RNA-seq is different. Next slide. So RNA-seq affords a number of important advantages in tumor characterization. For immuno-oncology, gene expression data is important for looking at which checkpoint genes and which neoantigens are being expressed. RNA can also give a more complete picture for gene fusions. And also potentially allele-specific expression and additional variant calls. Next slide. Perhaps more importantly, the integration of the DNA and RNA information together can allow for improved interpretation of the data, increasing or decreasing confidence in certain variant calls, for instance. So we have a whole session on this that Michael's chairing at ASHG. We're not going to have time to go through this in detail, but this is something that we think is very important. Next slide. And here's just a very simple example of how you can use this kind of combined information. So on the left here, you can see that you have healthy tissue as well as some renal cell carcinoma tissue where we've determined the copy number. And so for those samples where the copy number is abnormal, you can see that the expression also reflects, is also elevated, which kind of correlates or is concordant with what you'd expect with increased copy number. So anyways, a simple example, but this is the type of integration that can be very important. Next slide. Now, how is our ACE RNA-seq different? Well, for one, it's been optimized for cancer FFPE. So poly-A selection has been widely used for transcriptome sequencing, but its performance is limited when the RNA is degraded. On the other hand, ribosomal depletion methods suffer from inconsistent ribosomal RNA removal and a high percentage of reads mapping to intronic intergenic regions due to FFPE cross-linking. So what we've developed with our ACE RNA-seq is an exome or panel capture protocol based on our ACE technology, which overcomes these limitations and can produce high-quality transcriptome or targeted panel results from FFPE. We've also, acknowledging the fact that in cancer the samples are precious, that we've developed a DNA-RNA co-purification method that without affecting DNA yield. And this is especially important if you're dealing with limited tumor sample. Next slide. So what's the ACE impact on RNA performance? And you can see that it has a similar positive impact in terms of increasing the performance across different genes. And so this is a different type of blue-green plot. This is showing the RNA sequencing results for different genes. And these are all genes involved in cancer. VDAC1, for example, which is the second one down, the overexpression of VDAC1 has been associated with increased apoptotic response and anti-cancer drugs and treatment efficacy. And so the point here is that, again, the ACE approach really does fill in some of the gaps and really leads to a much more accurate and complete coverage for the genes that you're looking for. Okay, so before turning it over to Michael, I just want to briefly touch on the cancer content that we augment and target in our assays. So when we started the design of our assays, we looked to make sure that we had complete coverage over genes with clinical utility in cancer, consensus driver genes in cancer. We also wanted to make sure key cancer pathway genes, pharmacogenomics, and some immuno-oncology genes were also covered. And this resulted in over 1,300 cancer-related genes, another 200 microRNAs that are specifically augmented in our ACE cancer exome, and also these have become the basis for the large footprint of our ACE cancer panel. Next slide. So here's an example of some of the immuno-oncology genes that are covered in our exome. I mean, PD-L1 obviously is a very important one. You can see we have strong coverage in both these genes, and there are other genes like this relevant to immuno-oncology. The next slide. If you categorize these genes that we target by cancer type, you can see that we augment the exome and panel for genes that are involved both in solid and hematologic cancers, and we cover many more of the genes than other standard panels that are out there. Next slide. The cancer content that we target with the ACE cancer assays also includes more complete coverage of key cancer pathways, giving a more comprehensive view of the tumor biology. And this is important as you look to select patients with specific mutations for therapy. It's also important for evaluating response to therapy, especially when patients are receiving combination therapy where more than one pathway may be involved. Next slide. And the last point I want to make here is that we also detect variants in important regulatory regions. So this is an example. TERT promoter mutations are associated with aggressive traits of different types of cancers. We target regions like this that are important biomedically. And again, you can see that standard exomes and other panels typically miss these regions. Next slide. So in summary, regarding the assays, the ACE assays, standard sequencing approaches often leave gaps in coverage and hard-to-sequence regions that can impact neoantigen identification. What ACE technology enables is our ACE exome, cancer exome, and cancer panel to achieve best-in-class performance, ensuring that important neoantigens and variants are not missed. The ACE cancer exome and the cancer panel provide breadth of coverage needed for immuno-oncology applications. And the ACE cancer panel is the largest cancer panel available today. ACE RNA-seq is equally important and is optimized for cancer samples, thereby improving performance compared to standard approaches. And that RNA-seq is a critical part of the Immuno-Oncology neoantigen workflow. Okay. So with that, I'm going to turn it over to Dr. Michael Clark.
Thank you very much, Dr. Chen. As a reminder to
Well, thank you for the introduction. Next.
Yeah. As a reminder to our participants, if you have a question, you can type it into the Q&A box in the control panel, and we will conduct the Q&A after the presentation. So, Michael, you're next. Please proceed.
Thank you very much, Julia, and thank you, Dr. Chen, for the introduction. So now I'd like to talk about some of the applications of the ACE platform for cancer research with a special focus on how ACE cancer analysis and immuno-ID can be used in immuno-oncology clinical trials and translational research. Next. One area of great interest in personalized cancer therapy and cancer genomics right now is immuno-oncology. And immuno-oncology is a prime example of a cancer research area that benefits from comprehensive tumor genomic characterization. Before diving into how we use our ACE cancer solutions for immuno-oncology, I want to briefly discuss immuno-oncology as a field with you as it stands today. Next. So these figures show the basic mechanism of the immune checkpoint system, which I've highlighted with red boxes here. Immune checkpoints is a general term that refers to the numerous pathways that function to suppress and activate immune responses. These immune checkpoints are critical to the body's ability to regulate immune responses appropriately and prevent both collateral tissue damage from inflammation and autoimmunity. Now, the body's first and primary defense against cancer in general is the immune system. Through the recognition of neoantigens, which are mutated peptide sequences unique to the tumor, which the immune system recognizes as non-self, T cells are able to both directly attack tumor cells and to recruit the innate and adaptive immune systems to attack tumor cells. Therefore, there's a massive pressure on the tumor cells to evade the immune response. Research over the past few years has proven that some tumors can utilize the immune checkpoint system to evade the immune response. This therefore has led to a substantial interest in immune checkpoint blockade as a therapeutic approach. Next. Over the past few years, immune checkpoint blockade drugs have entered late-stage clinical trials, and some have even been FDA approved or received breakthrough therapy designations for some cancer indications. In the top left figure here, we can see that post-treatment with PD-1 blockade results in substantial infiltration of T cells into the tumor, leading to a therapeutic response. On the right, the figure shows some of the immune checkpoint ligands and receptors, many of which are being targeted by drugs in various clinical trials today. The major therapies that have been approved or that are in late-stage clinical trials are listed in the table on the lower left. And we can see that these target CTLA-4, PD-1, and PD-1's ligand, PD-L1. However, what this shows on the right is that there are a great many additional immune checkpoint targets left to investigate. Next. Now, with regards to therapeutic markers for immune checkpoint blockade, the expression of the target and ligand have been strongly associated with positive response to therapy. These figures show that the group of patients who respond to PD-1 therapy is enriched for PD-L1 expression by immunohistochemistry. In the bar chart on the top left, we can see in gray that patients who showed an objective response to PD-1 blockade also showed positive expression for PD-L1. This means that PD-L1 expression is a therapeutic indicator for increased response to PD-1 blockade. Next. So there's been a number of these studies now, and what they've largely demonstrated is not only the effectiveness of immune checkpoint blockade, but also the therapeutic value of the expression of immune checkpoint genes and ligands. Evidence shows that the more highly expressed these genes are, the more effective immune checkpoint blockade is likely to be in treating the patients. RNA-seq can be used to assess the expression of these genes, even on the single sample level, through comparison to a known set. And our ACE cancer RNA analysis that Dr. Chen mentioned earlier includes comprehensive expression results. Next. So the expression of immune checkpoint genes can be clearly seen in patient-derived tumor tissue samples, which is what I'm showing you here in this figure. And here we see 3 different patient-derived tumor tissue samples, including a basal cell carcinoma, which is a type of skin cancer, a pheochromocytoma, a cancer of the adrenals, and a renal cell carcinoma, a kidney cancer, as well as a control sample, which is a lab-grown cell line. All of these samples were sequenced on ACE and analyzed using our Personalis cancerase pipeline. The expression in TPM of the immune checkpoint genes is clearly elevated quite dramatically compared to the expression in the control samples. You can see each bar is actually higher than it is in the control. Moreover, we see that the expression of these genes varies from tumor to tumor. The basal cell carcinoma has substantially higher expression of PD-1, PD-L1, and PD-L2 than either of the other 2 tumors. This suggests that the patient with the basal cell carcinoma may respond more strongly to anti-PD-1 immunotherapy. For every RNA sequencing run we perform, we return data of this type so we can facilitate exactly this type of research and analysis. There's even more we can do with sequencing RNA and DNA with ACE. Next. So before I talk about some of the other applications, I want to talk a little bit about another major marker for immunotherapy response, the overall mutational burden of the tumor. In the top left plots here, you can see that patients with durable clinical benefit on pembrolizumab, which is an anti-PD-1 therapeutic, showed substantially higher counts of non-synonymous variants as a group than patients with no durable benefit. Further, in the bottom plots, you can see that progression-free survival on anti-PD-1 therapy was substantially greater in patients with higher mutational load. At first blush, this may seem counterintuitive, but the theory behind why this occurs makes a lot of sense. Essentially, a higher mutational load results in a higher quantity and variety of neoantigens, which the immune system recognizes as foreign and initiates an attack on. Presumably, this means that once the immune checkpoint blockade is initiated through immunotherapy, the T cells are able to mount a stronger response to the tumor and are more effective at killing it. Next. So looking at the same samples that I just showed you the expression of immune checkpoint genes in, we can see that they have substantially different quantities of somatic variants as well. By sequencing the DNA with ACE, we're able to call somatic variants in tumor samples. This gives us a sense for the mutational burden. The higher the number of somatic variants, the higher the mutational burden, and therefore, the higher the likelihood of having a positive response to immunotherapy. Now, something I didn't mention earlier was that these data come from a tumor normal paired analysis. In other words, we sequence both the tumor and the normal blood sample from these patients and then analyze them together in order to perform somatic variant calling. So I want to take just a moment to discuss how we handle samples with or without a paired normal sample in our analysis pipeline. Next. We actually have 2 different somatic variant calling pipelines, one for when there's a paired normal sample and one for when the tumor is being analyzed without a paired normal. In the tumor normal paired analysis shown on the left here, the normal sample is not simply used to subtract variants from the tumor variant set. Instead, the tumor and normal are processed together simultaneously to increase accuracy of somatic variant calls. Because this approach is very effective at identifying true somatic variants, we utilize a downstream filtering process catered specifically for the tumor normal somatic variant calls. When the normal sample is not available, we follow a flow shown on the right to call somatic variants. We utilize a substantially different somatic calling approach as well. This approach includes use of a panel of normal samples to reduce the chances of spurious somatic variant calls. Naturally, when calling somatic variants without a paired normal sample, private germline variants can actually be difficult to differentiate from somatic variants. For this reason, we use a different set of variant filters with our tumor-only somatic variants, and those filters increase specificity with a small trade-off in sensitivity at low variant allele frequencies. Next. So what I want to show you here in red is the total number of somatic variants called per sample when the tumor is analyzed in the absence of the normal sample. So it's pretty clear that the tumors ended up with a substantially higher somatic variant call number by tumor-only analysis. This is actually something that we see fairly consistently. Without the paired normal sample, because of the abundance of private germline variations, it can be hard to tell them apart from the somatic variations. But you can also see here that the number of variants differentiating a high mutational burden tumor like the BCC from lower mutational burdens like the PCC and RCC is smaller. That's the difference between the red peaks. And therefore, identifying a tumor that may respond more strongly to immunotherapy could be more challenging. Now, this data looks pretty striking and leads one to conclude that including the paired normal sample can be a very powerful approach. Next. So while this is true, I do want to point out that despite this, the number of actionable mutations detected here doesn't actually change between tumor-only and tumor-normal analysis. By actionable here, I'm referring to variants in the MyCancer Genome database, specifically variants that have a therapeutic indication for targeted therapy. Here I'm showing that across a large cohort of 79 cancer samples run either on the ACE cancer panel or ACE Exome, both with and without their paired normal sample, we noted no difference in the number of MyCancer Genome actionable variants detected. Across 79 samples, the same 25 actionable variants in MyCancer Genome were identified by both tumor-normal and tumor-only analysis. This means that although the overall somatic variant counts differ, depending on whether you use the paired normal, that difference does not yield actionable cancer variation. This actually makes a lot of sense if you think about it. So most of the extra variants in the tumor-only analysis are private germline mutations, and those are not likely to be actionable cancer variants. That said, the ability to differentiate high mutational burden versus low mutational burden is certainly improved by including the paired normal sample analysis. Next. I do want to mention that even though I don't have a lot of time to go into further depth on the importance of assaying the matched normal sample when sequencing cancer genomes, Dr. Elena Hellman from Personalis will be giving a talk on that topic at ASHG on October 10th at 10:30 a.m. So please attend that session if you're attending ASHG for more information. Next. So just to summarize this first part, the expression of immune checkpoint genes is a therapeutic marker for the effectiveness of immune checkpoint blockade. That expression can be determined on an individual sample by RNA sequencing, including targeted RNA sequencing with ACE. Also, the mutational burden of the tumor is another marker for immunotherapy. The higher the mutational burden, the more likely a positive response to immune checkpoint blockade. Mutational burden can be determined by DNA and RNA sequencing with ACE as well. Next. So in this next section, I want to talk a bit about a major emerging area in cancer immuno-oncology, neoantigens. So first, a little more background specifically on neoantigens. T cells have T cell receptors, TCRs, that interact with the major histocompatibility complex called HLA in humans on antigen-presenting cells and other cell types. But even tumors have to present antigens on their MHC in order to avoid the innate immune response that would occur if there wasn't enough signal from the antigen-presenting cells to the T cells. In general, just about any expressed peptide will be present as an antigen by the MHC to the T cells. When the T cells recognize an antigen as non-self, they initiate the immune response. Next. So those non-self antigens in tumors are called neoantigens. As we just discussed, tumors hijack the immune checkpoint system, which you can see represented by PD-L1 and CTLA-4 here, to prevent the T cells from initiating the immune response, which occurs upon seeing a neoantigen in the MHC. Next. So what specifically are neoantigens? Well, as we know, tumors typically have a number of somatic variants in their DNA sequence. These somatic variants are not present in the normal DNA from that individual. Next. Somatic variants can occur anywhere in the genome, but sometimes occur in a protein coding exon of a gene. Next. So these somatic variants from the DNA in the coding region may therefore be expressed in the RNA. Next. Particularly when the somatic variants are missent, this can result in the expression of mutated proteins with alternate peptide sequences. Next. So the peptide sequence that contains the variant amino acid will then be presented in the MHC as a neoantigen. This leads to an immune response when it's seen by the TCR on the T cells. This is the mechanism by which the immune system sees cancer cells as a target for attack. Next. So just going back to this slide, this may also explain why we see a greater benefit from immune checkpoint blockade in tumors that have a higher mutational burden. These tumors will typically also have a great number of neoantigens and therefore may have a stronger immune response. Next. So, coupled with immune checkpoint blockade, immunotherapy has started to take advantage of tumor neoantigens to create a stronger immune response in cancer patients through methods like cancer vaccines, as well as other methods. But I don't want to go into too much detail here on these types of methods, but cancer vaccines, for example, use neoantigens from the tumor cells to stimulate and activate T cells that will specifically seek out tumor cells expressing the neoantigen. Now, this has the potential, along with immune checkpoint blockade, to represent a major new branch of cancer therapeutics. Next. So, neoantigens represent a truly personalized cancer therapy. Every cancer patient will have a different set of somatic mutations in their genome, resulting in a unique set of mutated proteins and therefore a unique set of neoantigens. In other words, every patient has his or her own personalized set of therapeutic targets in the form of neoantigens. Next. So, one of the keys to neoantigen therapy is identifying that unique set of neoantigens in each cancer patient that can be targeted for immunotherapy. We have therefore created a sequencing platform built upon our ACE technology to identify candidate neoantigens from cancer samples called Immuno-ID. Next. So first, the cancer sample and the paired normal non-cancer sample from the patient are sequenced with the ACE cancer exome, which is augmented to optimize coverage, as Rich described earlier. Next. This paired sequencing is then analyzed through our paired somatic analysis pipeline. This generates a set of all the somatic variants that are predicted to cause a protein coding change that could ultimately result in a neoantigen. Next. Then we use RNA derived from the same sample that we got the DNA from to perform ACE transcriptome sequencing. ACE transcriptome features the same augmented design as the exome, but targets the RNA instead. Next. The somatic variants are called in the RNA as well as the DNA and then cross-referenced with the somatic variants between the two molecules. This gives us the set of all somatic variants that are actually expressed and therefore are the most likely to become neoantigens. Next. Now let's go briefly back to our basal cell carcinoma case and look at how ACE is used to identify neoantigens. So based on the paired tumor normal somatic analysis from ACE exome, you might remember that the basal cell carcinoma here had 222 somatic variants in DNA. Sequencing the RNA with ACE transcriptome revealed that just 44 of those 222 variants are expressed by the tumor. This demonstrates how powerful a filter the RNA sequencing can truly be. Next. So just to show some proof that the RNA sequencing enables us to accurately observe expressed variants leading to neoantigens, I want to show you the sequencing reads supporting these somatic calls in DNA and RNA. The sequencing reads from the DNA are represented by the light red tracks in the top half of this figure. The sequencing reads from the RNA are the light blue tracks in the bottom half. This particular somatic variant is a missense mutation in the coding region of the RPS2 gene, 127G to A. That's represented by the red line in the middle of the tracks in this figure. You can also see evidence that RPS2 has a high expression in this tissue, including expression of this coding variant. Also, there's evidence of splicing on the left side of the RNA track, right at the exon-intron boundary seen in the gene model in dark blue at the bottom of the slide, as expected. Next. Now, since ACE augmentation improves coverage over so many portions of the exome, it's natural that some of the potential neoantigens have their coding regions in areas that ACE improves compared to the standard exome. In this RPS2 example, the expressed somatic variant is present in a region that is benefiting dramatically from ACE augmentation. You can see that in the bottom figure, as the green area represents augmentation of coverage due to ACE, and the red line represents the location of the candidate neoantigen in RPS2. Next. So just to give you an idea of how this isn't really that rare, of the 44 expressed somatic variants that potentially lead to neoantigens from this particular sample, 23 of them are actually in regions that are substantially improved by ACE augmentation. Here I'm showing just a subset of those genes in the blue-green plot format. Every single one of these has at least some augmentation to improve coverage, including a few that have exons that would have been completely missed without using ACE. It's clear from these data that ACE exome and transcriptome identify substantially more additional true expressed somatic variants, and therefore it's substantially more sensitive for potential neoantigens than a standard platform. Next. So just to wrap this up, in conclusion, immunotherapy represents a major area of personalized cancer therapeutics. When studying immunotherapy, we need to be able to assess the expression of immune checkpoint genes, mutational burden of the tumor, and the identity of neoantigens. Exome and transcriptome sequencing enable us to assess all of these areas, as well as additional therapeutically relevant cancer variations. Personalis ACE augmentation has a tangible benefit for all these applications because it enables substantially more comprehensive assessment of these genomic elements. Next. So that just summarizes some of the applications we've been working on for Immuno-Oncology and the ACE ImmunoID platform. Thank you so much for listening, and I think we'd like to take some questions now, if possible.
Okay, thank you, Dr. Clark. Before we enter the Q&A, we'd like to ask attendees to please take a moment to take our exit service after the webinar has ended to give us your feedback. So now we will enter the Q&A portion of the webinar, and we had a couple of questions about the assays themselves. So what is your detection limit, and how deeply do you usually sequence for your cancer panel and the cancer exome?
Right. So, our detection limit on the cancer panel has been determined to be, for base substitutions, over 99% at a minor allele frequency of greater than 5%. For indels, a sensitivity of greater than 99% for minor allele frequency of greater than 10%. And then for copy number alterations, and that's a copy number greater than equal to 8 or 0, with the greater than 30% tumor nuclei, a sensitivity of 96%. And then for gene fusions, sensitivity of 95%, and the specificity overall of greater than 99% for those. So I guess the corollary to that question is sort of how we did the validation, which is, I think, very important, because the robustness of that validation really determines, sort of the quality of the actual metrics coming out of it. So one of the things that, one of the challenges in validation, I think, for the whole field, and this is something we've been talking to FDA about, is the lack of really good standardized samples that really begin to push the limits of detection kind of studies that you want to be able to do. So in the absence of those types of standardized samples, we've actually done our own work to create what we think are really robust ways to detect, to test the limits of detection. So, for example, for our validation for the panel, we procured 38 different cell lines, each with different small variants, a variety of copy number alterations, and also gene fusions. And for the SNP validation, we took 21 of those, mixed them into 15 different pools to generate minor allele frequencies, ranging from 5% and less than 5% to over 95%. And by using those different pools, we were able to determine our limits of detection. And so for indels, sort of a similar thing. If you actually look at the number of SNPs in indels that we end up with after you do all those pools, we end up with over 16,000 different SNPs at different minor allele frequencies and over 600 different indels. For copy number alterations, we did a similar type of thing. We did a dilution series to simulate reduced purity for the CNAs using these different cell lines. And for gene fusions, we also used known cell lines with known gene fusions to test our sensitivity and specificity for those. In terms of, and so the other part of this is that you also need to look at not just the cell lines, but also FFPE, because that's just a reality in cancer. So there are some standard FFPE samples out there, and we used those, and we did very well on those. I'd say that those are really not a great reflection of the reality of FFPE samples that are out there. to supplement that, we've looked at over 130 other FFPE samples from 20 different sites to get a variety of different FFPE as part of that, our studies on the FFPE and validation there. And that's the approach we've taken. I guess the other part of this is that, working with the FDA, and there was a question I saw that, regarding the FDA. Well, we think that the regulation of this space is actually quite important. And we've actually been working with them on the metrics and the parameters that should be established to measure performance for these different tests. And you can see kind of from our talk that we obsess about some of these, the details in terms of the coverage and the uniformity and sort of the sensitivity to variants. And things like, how well, not just are the genes covered, but what are they covered at? What percentage of the bases are actually being covered? Which exons are actually being defined in the gene definition or included in the gene definition? If there are hard to sequence regions, are those also getting covered? Those are all important parts of the analytical validity question. So we are working with the FDA. Deanna Church in particular has been meeting with them to help establish some of those metrics. We're using those metrics internally as part of our validation for our assays. And I think that's really helped us achieve high levels of sensitivity.
Okay, thank you very much. We have one question about single-cell sequencing. Can you do that with your platform?
So that's something we're investigating, but it's not commercially available.
Okay, someone else asked whether why you wouldn't just do the exome, why you have the panel and the exome.
Well, I think it's a great question. I think it has to do with there's a little bit of a trade-off between breadth and depth. So the exome has a larger footprint, and for neoantigens, I think that can be very helpful. And also, really characterizing the broad mutational spectrum for immuno-oncology. But because it's a larger footprint, that there's increased cost in going deep. And so the, typically for our cancer exomes, we're looking at 16 to maybe 24 G of sequencing. And that kind of gets you into the 150 to 200x depth of coverage, which is actually pretty good for detecting low allele frequency events. But with the panel, obviously, because it's a smaller footprint, you can go deeper than that, and so you have sensitivity for even lower minor allele frequencies than what you might get with the exome. Now, the trade-off there, obviously, is that with the panel, albeit large, ours is a very large panel, it's still not as comprehensive as the exome. And so I think, the exome and the transcriptome, the 8th transcriptome, we think are sort of the centerpiece of the ACMG solution. But I think it's important to put out there that we actually have another option for folks for specific applications where the large panel, and this is a very large panel, may be actually not just adequate, but actually ideal. So I think, there's two options out there, and there's that sort of sensitivity and breadth and depth kind of trade-off.
Okay, thank you very much. One question about turnaround time. What is your usual turnaround time?
Yeah, so for research samples, we're looking at 4 to 6 weeks, and for clinical related samples, kind of in 2 to 3 week time frame.
Great. Great, thank you so much. So we had a few questions about pricing information, and we will refer these to Personalis, who can address them individually with participants. This is unfortunately all the time we have for questions. We'd like to thank our panelists, Dr. Chen and Dr. Clark, and also our sponsor, Personalis. As I said, if we didn't have time to get you to your question today, we will have the panelists answer them directly afterwards. And as a reminder, please look out for the pop-up survey from the sponsor after you log out. Finally, if you missed any part of this webinar or wish to listen to it again, a link to an archived version will be emailed to all attendees. Thank you very much for joining us for this Genome webinar.